Good morning, everybody. My name's Dean Dawson. I'm the Senior Director and Business Manager for Nano IR Spectroscopy here at Bruker. And I'd like to welcome you to this Bruker Nano IR webinar. The presentation's titled New Advances in Nanoscale IR Spectroscopy. My presenter today, besides myself, is Dr. Curt Marcott, who is Senior Partner at Light Solutions. Curt is a former research fellow at Procter & Gamble. He's the 2011 president of the Applied Spectroscopy and is a member of the editorial advisory board of Applied Spectroscopy. He's currently an affiliated professor of materials science and engineering at the University of Delaware and an adjunct professor in the Department of Chemistry and Biochemistry at Miami University in Oxford, Ohio. And Curt obtained his PhD in chemistry from the University of Minnesota. So before we start with the presentation, I just want to make a few logistical comments. First, we highly encourage your participation during the webinar. So if you have a question, please type it into the questions dialog box on your screen. It should be towards the bottom right-hand side. So we'll accumulate these questions throughout the presentation, group them together, and then we can answer them at the end of the presentation itself. Quite often we have more questions than we can answer in the session, so in those cases we'll be sure to follow up with you via email afterwards with answers. Also, if you'd like to review the presentation afterwards or pass it along to a colleague, the webinar material and the link to the recording of this broadcast will be sent to you via email following the webinar. And then finally, when you exit the webinar software, you'll be asked to take a brief survey. And we'd very much appreciate it if you would take time to complete this, because it helps us select topics that are most important to you and relevant, and hope to generally make our webinar series a lot better. So, with that all said, I will go through the first part of the product presentation, which is the Nano IR3, and then after, and I will hand over to Curt afterwards. All right. Firstly, I just want to highlight that for those of you who may not be aware yet, that Anasys Instruments was acquired by Bruker Nano, so we are now part of the Bruker Nano family. That happened back in April 10th. So basically all the Nano IR products and support are integrated into the Bruker Nano family, and we're looking forward to lots of great product development that result out of this acquisition. So. All right, so just a little history for those of you who may not be aware of the history of Nano IR from the Anasys perspective. First Nano IR was introduced in 2010. It was first generation AFM-IR. And then subsequently over the years, we've developed multiple different generations of products, going from a bottom-up approach with the Nano IR first generation, top-down approach on the Nano IR 2, bringing in a new method called a resonance-enhanced spectroscopy. Then in 2015, we combined scattering SNARM and AFM-IR on one platform. And then in recent years, we've improved the speed and performance of the systems with new lasers and then also new modes such as Tapping AFM-IR. And then just recently, we've launched the Nano IR3. It's our latest generation Nano IR platform with second generation Tapping AFM-IR. So Anasys has a history of patented technology for Nano IR and materials property mapping. And Professor Alex Darzi from the University of Paris de Sud, who was the inventor of AFM-IR, won the 2014 Ernst Abbe Award. Right, so for those of you who may not be familiar about Nanoscale IR, just a quick overview of what it can bring you. So basically it gives you infrared chemical analysis at the nanoscale, sort of 10 nanometer levels. You can do chemical composition and nanoscale property mapping. Nano IR has been shown to direct directly correlate to transmission FTIR industry databases, due to its rich interpretable spectra. And then not only does it have high resolution, but it also has very high monolayer sensitivity. So just a brief overview of what its capabilities are. So the other thing is over the years, Nano IR has actually developed quite a broad range of applications, and later Curt will go through some of the latest advancements that have recently been published. But essentially it started out very much on polymer blends and polymeric materials and multi-layer films, but it's grown into a multitude of applications. Nanofibers, nanocomposites, organic nanocontaminants in the semiconductor and data storage industry. Life sciences has always been a prevalent application for it. And then also developing into 2D materials, graphene, and then also perovskites, etc. So an ever-growing range of applications is resulting from the developments of Nano IR. So coming back to the Nano IR3, we recently launched the Nano IR3 platform. So there are essentially 3 configurations of the Nano IR3. The first one is essentially a platform that's focused primarily on photothermal IR spectroscopy with Tapping AFM-IR. Gives high performance Nano IR spectroscopy. We'll go into that in a little bit more detail. Gives sub 10 nanometer resolution chemical imaging. And it has the same ease of use, high-speed measurements that you would expect from the result from the previous Nano IR platforms. Then the other options are a 3S for scattering SNOM imaging only. Allows you to do imaging in the visible, near IR, and other spectral ranges. And then the 3S is basically a system that has scattering SNOM and AFM-IR combined on the same platform. So it gives you the complementary capabilities of scattering SNOM and Tapping AFM-IR. And also allows you to do broadband spectroscopy and chemical imaging. So today, though, we are going to focus on the Nano IR3. And in future webinar, we will introduce the scattering SNOM capabilities of the system. All right. So just a quick overview of the Nano IR3. So next generation system has basically pushed its improvements in sensitivity for both spectroscopy and chemical imaging. We've made significant improvements in Tapping AFM-IR. We'll call it second generation. Tapping AFM-IR, and we'll go into that a little bit. We've introduced a new sort of Anasys Bruker branded IR laser, where we've worked with a third party to integrate lasers. And that is a hyperspectral IR laser, provides broader spectral range. So that's complementary to the other offerings that we have. It also provides sort of point spectroscopy, and each spectra can be sort of 1 to 2 seconds. Previous generation, it was 10 to 20 seconds for a spectra, so order of magnitude improvement in spectroscopy. And then we've retained the complementary material property mapping for electrical, mechanical, etc. And then, as mentioned, our ease of use, new AFM system enables us to generate improved high performance images with our maintaining the Nano IR ease of use. So just an explanation of what are the key areas of the Nano IR3 that we've improved. So firstly, from the top, we basically have now a fully purged system. So this is important for removing background from the IR signal. So from the delivery of the laser to the sample, the whole system now is fully purged. Previous generations did not have that capability. Then we also have the second generation Tapping AFM-IR, lots of significant improvements. I have a separate slide on that. We'll go through that separately. We've improved our optics train and improved our beam pointing design, improved the density of the IR beam underneath the probe, all leading to improved signal to noise. We've also done a significant redesign on the AFM. So we've reduced the noise to 50 picometers. In an acoustic code, it can actually go lower. And this is basically down to a new head and scanner design that we've incorporated into the system. And reducing AFM noise directly correlates to improved IR signal to noise. The system also supports AFM-IR and scattering SNUM. So it's if you are interested in scattering SNUM or AFM-IR, you buy one option or the other. The other option is easily field upgradable. So this provides much broader experimental flexibility that's built into the system. And then we also have the new hyperspectral laser and also other laser options that enable us to cover a wide spectral range from below 800 wave numbers to 4,000 wave numbers for both scattering SNUM and AFM-IR. So we talked about hyperspectral imaging. This is an example of the capabilities that the Nano IR provides with the new laser. We've been providing hyperspectral on the previous generation. We've now brought out a more fully fleshed out product, file formats for hyperspectral imaging, and which are exported to external analysis packages that can be analyzed by the users. So an example here is the 5 micron by 5 micron 50 by 50 spectrum array on a standard sample that those of you who have a Nano IR will recognize it immediately. So it's powerful capability. It's essentially a great way for you to look at the whole of the sample across a broad array, and is being used quite significantly by our Nano IR users. So a couple of other things, not just the product, but also we're fleshing out a lot of the accessories. Firstly, we have sample environmental control that can be available as options. Firstly, we have humidity control and a heater cooler that allows for in situ AFM-IR as you control the humidity or use another gas of the sample, as well as if you're heating and cooling the sample. So this is, again, important thing here is it's in situ AFM-IR for humidity control. Another new capability that we're offering on the 3S is a sample transfer port. So example, 2D materials, getting prepared in a glove box. You want to protect the sample, make sure there's no oxidation effects when you measure it. So this sample can be taken from the humidity box, from the glove box into into a sample transfer mechanism, which is then put into the Nano IR system through this sample transfer mechanism here, and then you can go on and measure the system. Measure the sample in a protected environment, as the whole environment within this cover can be filled with argon gas or other gases that may be important for the application. So we have, to enable control of the environment, we have humidity sensors, optional oxygen sensors as well. So this is a unique capability we added specifically for 2D materials, but then also lots of other applications could warrant this level of this type of sample protection. So the other thing that's important, and actually Curt will be going into this in a bit more detail, is polarization control. It's a very useful capability. Recently, as a paper released giving numerous examples of polarization control. So this basically allows you to study the molecular orientation by changing the input polarization of the IR light, whilst studying the changes that go on in the sample. And then also we have extended, one example of a laser that we have for extended range up to 4,000 wave numbers is a fast spectra, but we also have other lasers we're developing that we'll talk about in future webinars. So then we've also retained our nanoscale property mapping modes. Conducting AFM, KPFM, and also Nano TA and the LCR. Those modes are also available on the Nano IR 3 as they were before on the previous platforms. So then just coming to the second generation Tapping AFM-IR. So essentially we've improved Tapping AFM-IR through a number of ways. Firstly, just improving the AFM design, improving the noise floor directly improves the performance of any type of measurement we do, IR measurement, including tapping and resonance enhanced. The improved optics design is looking to remove the background signals, etc. But we also have new AFM probes, new Tapping AFM-IR algorithms that we've applied. And so a combination of these factors have enabled us to improve the sensitivity, the general resolution, spatial resolution, and then also what we've been able to add now to second generation is really enhanced spectroscopy capabilities. The first generation was primarily in imaging mode. Now, Tapping AFM-IR can be used for both imaging and spectroscopy. So it's a significant enhancement to Tapping AFM-IR, and a number of these improvements are available for upgrade to current users, with the exception, of course, of hardware things like the AFM. And a good example here is we took an image of a graphene wedge on silicon surface, and this is SNARM absorption and reflection images. And what we see here with Tapping AFM-IR is we are able to image the plasmonic effect at the edge of the wedge using Tapping AFM-IR, which sort of points towards its improved sensitivity of the mode. So this is the first time we think that we've seen plasmonics on a graphene wedge on the silicon. So that was a general overview of the Nano IR 3. What I'm going to do is I'm going to hand over the presentation to Curt, and he will then show you some new Tapping AFM-IR data, and also go through lots of new capabilities, papers and research papers that have been recently published this year. So just bear with me while I make Curt the presenter. So Curt, you should be able to present now.
Okay. See if I got this. All right, how's that, Dean? Is it looking right?
Looks good.
Let's see.
Yes, just need to put it in.
There we go. How's that?
There you go. That's great.
Okay. Thanks, Dean. So before I get into some recent publications, I want to give a couple of examples of the new Tapping AFM-IR. The first one is a blend of polyethyl methacrylate and polymethyl methacrylate. So these are very similar polymers that have very similar spectra. And on the left-hand side here, you can see the tapping AFM-IR spectra of the two materials. And if you look at 1026 reciprocal centimeters, you can see that the PEMA has a band there that's much stronger than the PMMA. So that means now if we do mapping and tune the IR laser to 1026 reciprocal centimeters, we can image and you are able to determine that the continuous domain is the PEMA, and the domain areas, the blue areas, are the PMMA in the image. We can also overlay this into a 3D image that overlays the height with the chemistry. So you can see the islands of PMMA in the sea of PEMA using the tapping AFM-IR. This sample came from the University of Minnesota. The second example is even smaller domains. This is a block copolymer of polystyrene and polymethyl methacrylate. So the domains are extremely small. The AFM image here is 2 microns by 2 microns, and then where we zoomed in and did the tapping AFM-IR measurements is 1 micron by 1 micron. You can see the 250 nanometer scale bar. So if we tune the IR laser to 1730, that's a band of PMMA. There's no carbonyl and polystyrene. We get the image shown here where the lighter colored areas are the PMMA in the sea of polystyrene. And if we look at the line here drawn through one of the domains, you can see that we've achieved 5 nanometer spatial resolution in the tapping AFM-IR image of this material. So those are 2 examples of AFM-IR. Sure, we'll see many more published now as this capability propagates into our user base. I wanted to go through some publications, and I started looking at them, this is just publications from 2017 and the first half of 2018, so basically 18 months. And it's only a sampling. There's well over double the number of applications out there. But our users are really now starting to generate some very nice work using the AFM-IR technique. And so I'm going to go through 4 of them in the life science area and 4 of them in the material science area, and then I'll end with a paper on atmospheric aerosols and polarized AFM-IR. So there's a recent paper in Cell, which is one of the highest impact biojournals out there, where they use Nano IR, AFM-IR to determine the binding of these RNA binding proteins, and they could vary the structure of these proteins depending on whether they methylated this arginine component or not. So they formed 3 different FUS, which is the RNA binding protein structures. And one of the key things that I like about the AFM-IR technique is you can get information about the morphology and the chemistry from the IR spectra, but you also at the same time get information about the mechanical stiffness. So if you look at these 3 examples of different types of RNA binding proteins that are produced, you can see that they have stiffness variations and they have spectral variations that show up. And you can map the relative stiffnesses of the 3 different types of RNA binding proteins. And in this one in the middle in particular, the hypo-FUS, you can see that there's a region where there's a sphere and a region where there's an elongated area, and the spectra are different from those 2 areas of the sample. So you can actually get down inside of these materials this is like a 5 micron scale bar, and get information about the stiffness as well as the chemistry. And if we overlay the spectra now, the co-average spectra from the different domains, you can see that the signal to noise is good enough that you can do second derivative spectroscopy of the amide-1 band and get more information about the proteins there. So the bottom line is you can get information about the mechanical, secondary, and quaternary structure properties of individual FUS condensates and get substantial differences depending on the way they're set up to bind. There's a recent paper that's just been accepted online in ACS Nano from Andrea Santoni's group at NIST, where they've been able to determine polypeptide conformation in water with nanoscale resolution. And there's a lot of people that would like to be able to do AFM-IR in water. Now that's been difficult. There's some work out of Texas A&M, out of Mikhail Belkin's group, where they first were able to do this, and they have another recent paper. But this paper, it just accepted and will be appearing soon in ACS Nano, shows what the problems are. One of the problems, of course, is that water absorbs very strongly in the IR, and so we move back to the Nano IR 1 configuration when we come up through the bottom with the laser through a zinc selenide prism. And the reason is we get less depth of penetration into the sample, and that helps us reduce the water background. The other thing is, though, the water covers not only the sample, but it covers the AFM tip and cantilever. And when it does that, if you compare the cantilever ring down frequencies for a sample in air versus a sample in water, you can see there's a big shift to lower frequency when you put these things in water. The cantilever gets damped down and the amplitudes are smaller. So that's another one of the problems that you need to overcome. So the ability to do resonance enhanced AFM-IR is crucial, where you map the rep rate of this laser to be right on top of one of those cantilever ring downs. That can give you enough sensitivity. And here's an example in water, H2O, of some phenylalanine fibers. Now these phenylalanine groups are models for plaque-forming fibrils structures that occur in proteins. And so we compare at the top the AFM-IR spectra in air to the spectra in water, and you can see there's differences because the molecular conformations and so forth change quite a bit. So the diphenylalanine is the core recognition module of the Alzheimer's disease beta amyloid polypeptide. And again, we can do second derivative spectroscopy, and we can see differences between the spectra in air and water. We can also move to D2O and now compare to this tert-butyl-carbonyl modified structure. This one is similar to the diphenylalanine, except it's got these other groups put on it, and that prevents it from forming beta sheets. And so the experiment's a little easier in D2O because you move the water absorption band that normally overlaps with the amide 1 band out of the picture, and you can see here that for the Bach FF in D2O, we see the alpha helical form there, and when we move to the FF without the Bach group, it forms the anti-parallel beta sheet. And so the signal to noise is a lot better in D2O, and we can differentiate between anti-parallel beta sheet and alpha helices in these materials in water. So I think this is opening up the ability to do the experiment in water, something people have wanted to do for a while, and that's starting to become close to a reality, and this is a really nice paper. I'm not going to have time to go through a lot of detail on these papers, but the references are there, and you can go, if you find something particularly interesting, you can go back and look at it in more detail. This is the first of 2 papers that just came out of Baden Woods group at Monash University. They're now doing AFM-IR of malaria-infected blood cells. And so now we can get inside of single blood cells. So this is an organelle inside of a blood cell, and you can see the spectra that were collected from outside that area and inside that area are different. And you can do a principal component analysis and make images that differentiate where the material inside the cell is from outside the cell. You can see differences as well in the amide 1 band. And these are reference spectra of different protein materials that might be found inside the cell. The second example out of Baden Woods group is AFM-IR spectroscopy of bacteria. And so they were actually able to collect IR spectra of a cell that's been in the process of dividing. And you can see a spectrum collected right on the septum where the cell is dividing compared to a spectrum taken away from it. You can see these bands down here around 1200 wave numbers that are different. These are due to phosphonate and skeletal modes that are starting to pop up right on this membrane. And they were able to show after the experiment that they were able to make these cells come back to life by rehydrating them. So it's not really it's an in vivo measurement of a bacteria that was later shown to still be viable after the experiment. So that's another example of the kind of work you can do now with AFM-IR. You can now get inside single cells and look at organelles. This is some work that was just published out of Dow by Mark Rickard and Greg Myers there. They've been able to look at a deuterium-labeled polyolefin copolymer in a polyolefin blend by AFM-IR. And so the polymer they're looking at is a blend of polypropylene and polyethylene, and it's sort of compatibilized with a ethylene propylene copolymer. And what they did is they formed the material, and it forms domains. You can see this is about a 20 micron by 20 micron area. And if you take an IR image at 1377 reciprocal centimeters, you're lighting up the polypropylene rich phase. And if you switch to 1473, you light up the polyethylene rich domain. And what you really want to know is, where's this ethylene propylene polymer that compatibilizes the two? Where does that occur? If you ratio these two images, you get a much crisper image, because 1473 contains both methyl and methylene contributions, while 1377 is mainly from the propyl. So you can see where the polypropylene layers are. So if we look now at the deuterated material, you can see the CD stretches up here have now shifted from being around 2900 to being around 2200. And this is the ethylene propylene polymer that's been deuterated. And then if we look at the spectra now, this is the AFM image from outside the area where the polyethylene is predominant. We see evidence of the deuterium material, and we don't see any evidence of it in the polypropylene material. So this is telling us that most of the deuterated EP component that was added in is going into the polyethylene domain. So this is all work out of Dow. This is some work that is out of the University of Delaware that was performed by Leon Gong, who was a graduate student there at the time and is now at 3M. And this is pretty exciting work where you've been able to look at electrospun fibers that are only like 250 nanometers in diameter. And not only can we get infrared spectra of these individual fibers, we can see a difference between the infrared spectra in the core of the fiber versus the shell. And if you image at the high wave number component of the PHB copolymer carbonyl band, we see that the outer layer of the sample, the shell of the sample, which is only 9 nanometers across, lights up as being strongly absorbing. So this paper, which just appeared in Macromolecules last year, shows that the core of these PHB copolymer fiber is mainly helical, but on the outside is this 10 nanometer thick shell. And not only can we light up that layer, if we use polarized light, we only see the carbonyl when the light is polarized perpendicular to the fiber. So we can determine as well that the shell is made up of beta-like chains where the carbonyl groups all point away from perpendicular to the fiber core. Normally you think of graphene as being something that's difficult to do with IR. That's true if you just have regular old graphene. You don't really get an IR spectrum. Raman works much better. But this is a paper that just came out of Suzanne Morse's work at Manchester University, where they were able to look at functionalized graphene. And so I think this is an example where AFM-IR can complement Raman in getting information about the functionalization of the graphene, because Raman can't really see that. All they can tell is the main graphene structure. They can't really tell where the functionalization is. And so these are two different functionalized graphenes that they made. The regular graphene, the URGO, has no functionalization at all. And then they have two different functionalized forms that are only slightly structurally different. The PBS RGO and the PC-NBS RGO versions. And so they used AFM-IR, and they could get spectra that showed when they looked at the unfunctionalized graphene, it's flat, they don't see anything, but they get spectra when the material is functionalized, and they can collect IR spectra from different locations on the sample and get information about the functionalization of the graphene, and they can even do imaging. The regular old graphene, they don't see any IR images at all. with the one functionalization, they light up where the functionalized areas are, and with a different functionalization, they can do that as well. So you can get information that complements what you might get from Raman about the basic graphene structure. You can get information about the functionalization and the location of the different functional groups on graphene. Normally, we don't think of photothermal AFM-IR as being that useful for 2D materials. Dean showed that example of the graphene tip where they were able to see the photothermal effect on the end. This is another paper published on data collected at Harvard, where the main point of the paper is they're able to see a large photothermal effect on a sub 40 nanometer thick boron hydride material. So in this case, in the thinner regions, at different points in the sample, off the sample they don't get any signal, they get spectra of the silicon, and then when they move into the thicker regions they can see this band on the shoulder. And so if they do a photothermal image with the laser tuned to 1368 reciprocal centimeters, they light up the thicker layer of the boron hydrate, and they don't, if they move to a wave number where it doesn't absorb, you don't see the contrast. So it is possible to do photothermal IR spectroscopy and imaging on 2D materials with AFM-IR. It's also a first application that just came out in related to geoscience, looking at coal, shale, and things like that, looking for organic materials. This is work done in collaboration between Schlumberger and the U.S. Geological Survey. And in this case, if you look at a piece of coal, the scale bar here is 10 microns. You see different areas of different types of coal with different maturity levels. And if we map out, you get, and as I mentioned before, you can get information about the stiffness. And so you can isolate areas of the sample and collect spectra from the different locations of these three different types of coal. This is the solid bitumen, and this is tasmanites, and the three different areas. And you can get distribution maps of the stiffness, and you can see the functional groups are different. The infrared spectra are different depending on the age of the maturity of the organic material that's being formed in the shale. This is a paper that just came out in analytical chemistry out of Mark Benezek Hall's group at Michigan. Mark has since moved to Monash University, and he'll be getting a Nano IR system set up there very shortly. But he's been able to show that he can deposit individual particles from an aerosol and do AFM-IR spectroscopy to identify chemically what these particles are, and they're on the order of 100 nanometer spatial resolution, single particle spectra. Just last month in Applied Spectroscopy, a focal point review article was published by Karsten Hinrich's group. He was the first Nano IR customer in Germany, and he's really been doing some extremely nice work taking advantage of the polarization. This is an area that's dear to my heart. I've always worked with polarized IR, and I think it's an area that really a lot of opportunity for AFM-IR if you take advantage of the fact that the light is polarized. And so this paper has a whole bunch of examples. It has a polarizer that you can rotate and get light polarized parallel or perpendicular to the sample, and then by knowing the transition moment directions of the vibrations, you can get information not only about the chemical structure of the material on your sample, but you can get information about the molecular orientation. So the one example in that review paper, there's many more, are these aggregate material, peptide materials, and you can see they form different structures. This is a side view of one of the helical structures that formed, and then a top view, and then there's a second structure that has sort of a tiered location. If you turn that 90 degrees, it's linear. And if you measure the spectra with both p-polarized light, which is light polarized perpendicular to the plane of incidence, and s-polarized light, you can see these two structures are dramatically different, and we get a different spectrum depending on how the light is oriented relative to the substrate. And so you can make maps of the IR images at different wave numbers and get information about the molecular orientation. I encourage you to take a look at this paper that just came out in the June issue of Applied Spectroscopy. There's many more examples about how you can use polarized light to get nanoscale molecular orientation information. I want to call your attention to the SciEx conference. It's turned out to be really a meeting place for people doing nanoscale IR and also Raman spectroscopy. And for like the third or fourth year in a row now, we're going to have a session where we have alternate talks of AFM-IR and nanoscale Raman spectroscopy, nanoscale IR and Raman. There's still time to submit a poster for that conference, which is held in Atlanta. So those of you in Europe, that's a pretty easy place to get to. And there's a whole series of talks at that conference. These are all speakers that are speaking in sessions that I'm going to be chairing. Some of the work I've already talked about a bit. Andrea Centroni will be talking about peptide conformations in water. Simone Ruggieri will be talking about identification of structural characteristics and inclusions in neurodegeneration examples. Karsten Hinrich, who just did the polarized result, will give an overview talk on how to do use AFM-IR to do anisotropy of thin films. Mark Ricard, I showed some of his work on the polyolefin blends. He's going to be talking about some new work on flexible packaging films. Karen Weiland is from Vienna, and she gave a talk that got an award for a graduate student with Bernard Lendo, got an award for one of the best talks at SciEx, Spring SciEx in Glasgow a couple months ago. And so we invited her to come and talk about this work, assessing drug capsulation in liposomal nanocarriers. Centroni's group is also co-authors on this paper. It's a really nice work, so we're glad that she's able to come over. And Alex Dazi's graduate student is going to talk about PTIR spectroscopy of organic matter in Antarctic meteorites. Then there's going to be a talk by Ariana Denisset on overviews of photothermal induced IR. She's from University of Paris Sud. And then Mark Banachek-Hall has a new paper, some very interesting new work that he was invited to speak about in a special session on nanoplastics, where he's been able to use AFM-IR to detect nanoplastics in seafood. This is a really important area, and I think an important one in the future for AFM-IR to be able to identify nanoplastics which are appearing everywhere and are a serious problem. I also want to point out that coming up in September, this is actually before the SciEx meeting, there's going to be the third annual European Forum on Nanoscale Spectroscopy. This has been a very successful conference that Anasys has been running. This is for the third time now. Dean's been very much involved in getting this thing going. This year it'll be co-hosted with the National Physical Laboratory in London, outside London, and it's a good opportunity to share cutting-edge research and ideas in nanoscale research, and there's still opportunities to submit posters. If you go to the Bruker website link shown there, you can see more details, and there's still time to submit a poster. And here are some of the invited speakers for that meeting. A lot of the work that I talked about, these people were co-authors on. Simone Ruggieri will talk about his work in fluids, AFM-IR, and cells. Suzanne Morsch, who did the work I talked about in the application section, will talk about AFM-IR of solid insulation. Tom Hoffman, Professor Wojciech from Poland. And then there'll be a whole lot of talks there, and you can go to contact Dean, especially for more information about this conference. So in summary, Nano IR spectroscopy and imaging performance improvements have improved continually and enable us to access a much broader range of applications. The data get better and better, and as excellent scientists now get this tool in their hands, they're generating increasingly interesting scientific results that haven't been able to be obtained in the before. Some examples in this webinar show that Nano IR spectroscopy provides unique data sets and new research insights that are not available with other techniques. And nano IR technology continues to grow and improve spectroscopy signal to noise, chemical spatial resolution, while improving surface sensitivity and becoming broadly adaptable. So with that, I'll stop and we'll take some time to entertain your questions. And I'll turn it back over to Dean.
Okay. Actually, just leave that slide up. That'll be great. We have a ton of questions. I guess just bear with me a minute. So there's a couple of questions on fluid AFM-IR. So I just got to find them. I think the first question was, just give me one sec. Well, the first question was, for fluid AFM-IR, does Bruker Anasys have a component to allow for fluid experiments? So a lot of the fluid work, maybe Curt, you can just explain briefly, because I think the other question was related to how do they do fluid measurements in AFM-IR, and wouldn't that impact the signal? Maybe if you just briefly explain the principle they used, that Simone and Andrea used.
Okay, first of all, you have to go back to the first generation instrument, the Nano IR1. No one's doing fluid on a top-down. Everyone who's been successful has gone up through the bottom through a zinc selenide prism. And I believe Andreas Antoni was our first Nano IR customer. He bought the first instrument back in 2010, and that was the one he had. And he still uses it all the time and is publishing papers on it. And Simone Ruggieri, who was at EPFL, cut his teeth on the Nano IR1 also, so he's very familiar with that. And Mikkel Belkin, his instrument at Texas, he's the other guy doing that sort of work, as well as Alex Stasi is working on it. They're all using the bottom-up approach. So I don't think there's anything that Anasys supplied them to make that experiment work in hot water. I haven't seen their instruments to know exactly how they're doing it, but that's what it's going to take. And as I give talks all over the world, that there's a tremendous amount of interest in trying to do this in fluid, and I think it's becoming close to a reality. And perhaps Bruker now can make something out of the old prism bottom-up approach to be able to do this more effectively. But I think the users that are doing it to date have pretty much taken their Nano IR1s and modified them on their own.
Right. Yeah. So we are looking at trying to incorporate that type of concept into the Nano IR3 as a bottom-up only, so that it's sort of a newer generation, takes advantage of the 3 platform. But that's that's sort of a next year development. But it's definitely an area where clearly we're seeing researchers do work in fluid. There's lots of interest in it. So we're looking to definitely support that. So, so then there was a question on the cell measurement. I don't know. It says, to have a spectral comparison for the inside and outside of the cell, do you need to cross-section the cell?
I don't believe you need to cross-section the cell in that case. I don't think that was done at all. Right. The cell was thin enough that it didn't need to be done. Okay. After it was dried, and so it gets a lot thinner in that case. I see Simone Ruggieri's online. He probably knows the answer to it since he made the measurement.
There you go. Then there's some questions about resolution. So the first one is, does 10 nanometer scale mean that spherical nanoparticles of 10 nanometer would be detectable and could be analyzed by IR? Well, so our definition of spectral resolution for nano IR imaging is similar to how you would do it with an AFM. All right, so it's essentially detecting the change in a feature transition. However, that said, we are able to measure smaller and smaller features. A good example is some organic nanocontaminants are becoming not just very thin, but also essentially they're particles. So we're doing work there measuring very small particles, certainly not 10 nanometer by 10 nanometer, but not much larger than that. So the strict definition is transition from one feature to another, but yes, we can measure small sort of particle-like features. So then there's a question about it says for your older model Nano IR2, the resolution of the chemical imaging is limited by the AFM tip. It says, I think basically it's saying is the resolution of AFM-IR limited by the tip radius and the laser spot size? So did you want to talk to that? Or?
Yes. It's not dependent on the laser spot size, because the laser,
Correct.
You can't really focus it smaller than about 20 microns. And so the key is the tip. Ideally, that would be the limiting spatial resolution, the diameter of the tip. In practice, you don't always achieve that depending on the sample and how fast it dissipates the heat after it's excited, or based on how thick it is. So to get the best spatial resolution, the sample typically is very thin to achieve the best spatial resolution. But ideally it's the diameter of the tip, but that's, in the most favorable cases, usually it's somewhat less than that. And a lot of the contrast we're getting, particularly with tapping AFM-IR, is related to stiffness differences. And those things you can scan a lot more finely. And then you use the spectral information to verify that the two materials are what you think they are. But a lot of the contrast that's coming is just because when you move from one type of domain to another, the stiffness changes, and the tapping AFM-IR is very sensitive to that.
Great. So then there are some questions about simultaneous measurement. It says, can Nano IR3 and AFM-IR and SNOM, can it do them simultaneously? So, I mean, the short answer is no, but you can. We actually have a probe that we're testing to be able to switch between AFM-IR and scattering SNOM to allow you to do, so you're measuring the same location on the sample, and you can look at the, reflective and absorptive components that the two modes give you. So that's the answer to that. Then there's some general questions on lasers. So there's I guess the best way to answer this is there's a lot of work going on with lasers. So we're working with a number of mid-IR laser manufacturers to make sure that the spectral range and the performance and the price also improve in all vectors. And I would, basically we're trying to cover the range from sort of 650, 670 range up to 4,000. That's the sort of broad range that we're trying to address. So at the moment you need a couple of different lasers to get to there. The new laser that we've introduced goes from sort of just below 800 wave numbers to 1800 wave numbers. Then you need to add another laser if you want to extend to higher wave numbers. So I would say if you have a question, or you have an application that requires a broader range in different parts of the mid-IR range, then I think just feel free to reach out to us because there's definitely developments in that area that we are working on, and we will in fact have a special webinar specifically to address the 3S, and then also a new laser development that we're working on there. So yeah.
I would just like to add that to get a different spectral range with the Nano IR products, it's all about the laser. If a better laser technology comes along, we can immediately take advantage of it and make the instrument a ton better. And you don't have to buy a new detector because your AFM tip is your detector. So we're in a position to take advantage of any advances in laser technology that may come along. And laser manufacturers are always coming to Bruker and Anasys with their newest laser to see if it'll work on this experiment.
Yeah, and there was some questions there specifically. Can we get the hyperspectral laser on the previous generation products? Yes, you can. In principle, most of the lasers that we work with or develop are backward compatible to our current Nano IR systems. May require a little bit of integration, but they're definitely compatible. So then there's a question on temperature control. Is it sample stage or the entire sample chamber? So we're controlling just the sample itself, not the entire chamber. So that's a quick answer to that one. Then, okay. So then there's a question about, please explain sensitivity of different resonant modes and cantilever length effect. I think that actually might be a bit much to answer in this. That's probably a long answer. And there's some general questions. So sorry, I'll have to send an email response to that. So there's some questions about general availability of the instrument. So we are shipping our first Nano IR3s this month, actually, at the end of this month. So it's available. And a lot of the new improvements, just to comment for those who do have Nano IR, I mean, a lot of the new improvements that we've added on Nano IR3 are backward compatible. Clearly, if it's a new AFM design, it's not. But the environmental chambers and heater coolers are available for the Nano IR2. The improvements, as much as we can roll them back, for the improvements for Tapping AFM-IR are available on the previous products as well. So it's if you're interested in some of those improvements, then just get in contact with myself or your local Bruker salesperson. So we are coming up to the hour. There was a number of questions that we didn't get to. So we will definitely respond to them through email. A number of you have sent multiple questions. So for sure, we'll definitely follow up. Okay. All right. So I think that concludes today's webinar. I'd just like to thank everybody for attending. And please do fill out the survey. We are interested in your feedback, and we will for sure be following up with you on responding to the questions that we were not able to get to today. So thanks again, Curt, for all of the presentation that you gave, and thanks everybody for attending. Goodbye.